Okay, so in the second module, there is a PowerPoint that goes through like Immunoblot basics. I'm going to use that to give you a little bit of that information. It talks about all kinds of amino acids, by the way. It will talk about, ELISA and immunohistochemistry and Western blot. So you can kind of learn about all three things in there, but I'm gonna just kind of focus on the Western blot portion. Now remember, Westerns are, semi-quantitative. So because it's just a band, right? And you're looking at the band intensity, even in the best case scenario where we use ImageJ or another method to quantify, like the amount of signal there, it's still going to be relative to each other, the samples that are present on that particular membrane. You can't compare from that membrane to a different membrane. It's very hard to do. So it's semi-quantitative, and it can just tell you relative amount of your favorite protein within the different samples present on that lot that you're looking at that time. Um, for the sample buffer, for the samples, because Western blots can be used to visualize and to look at the relative amount of protein, full length protein, as well as very specific like phosphorylations or glycosylations, if you have good antibodies against them. When you're extracting the protein, it's imperative that if we are interested in those uh, post-translational modifications, that we add phosphatase and glycosylation inhibitors to the lysis buffer and preserve them. In addition, we always add protease inhibitors to it, so the proteases that are present inside our cells don't just chew each other, all the other proteins up, and we can get actual examination of the proteins. Another thing with Westerns is for best results and for best kind of quantitation, so-called, of our samples, and for best comparison between samples, what you want to do is quantify your proteins in each of your extracts that you're going to be using in a Western blot and equalize the samples so that they can be really compared to, right? So you use 100 molecules of protein in each of your samples, for example, right? Or 1 microgram of protein or 10 microgram of protein. You want to load equal amount of sample in each one of your lanes so that you can actually make good conclusions about your favorite protein. When we are running the gel, again, it is important that we choose the right type of gel. They're all either Tris HCl or acrylamide gels, whatever they are. They're going to be a different person. You can have different percentages depending on the range of molecular weights that you're interested in looking at. You want to choose the gel that is appropriate for that. So for example, if all the proteins, if I have 3 proteins and all my proteins are high molecular weight, 100 and above, then I would want to load them on a low percentage gel. So because I don't really care about the lower end molecular weight end, right? I don't care about those small proteins. I'm only interested in those large ones. So I want to separate them as much as possible, make them nice and clean and separated so I can look for my 3 individual proteins in that range easily. Because look at this, if instead I were to use a 12% gel, which is typically what people use because it gives the widest range of proteins to look at, uh, that section is really scrunched up. So it's going to be really hard to separate those 3 different areas, right? 3 different protein bands. So to resolve those high molecular weight proteins, I would want to use a lower percentage gel. If, on the other hand, the proteins that I'm interested in are more, in the middle range, then I want to go to a higher percentage, like 10% or even 7.5, depending on which range I'm looking at, right? If we have proteins that are all over the molecular weight range, I have some super tiny ones and some really big ones and some middle range ones. What we can run instead are what we call gradient gels. 4 to 15 is my favorite, by the way, just because it gets both the high molecular weight and some really tiny proteins well resolved. You do have to kind of keep an eye on the lower side to make sure that you don't lose these 10 to 15 kilodalton ones. But in general, it resolves really, really nicely for all molecular weight ranges. Another one that I like to use a lot is the 10% one when I'm running cyclins or cell cycle proteins, because a lot of them fall between 75 and 25. And so by doing this, basically this resolves that area really, really well. So I can cut my membrane into various parts and look at all my cyclins at the same time on the same membrane. So if you choose your gel wisely, you can run even 10 different, antibodies on it, or at least, 5 or 6 antibodies on it easily without having to even reprobe. You can cut out different areas of molecular weight range, put them in separate vessels and add different antibodies to them appropriate to that area and look at them. And that works out super well. Another thing we can play around with are different buffers. You just need to know that you can adjust the buffers, pH and stuff so that your gels run faster or better depending on what it is that you're looking at. And these buffers are always changing as they're getting developed by different companies. So we always look at whenever we are running Westons, after a while, we will always look at the different options available and see what would be best for us. So today it's MES and MOPS, tomorrow it's going to be different, but the basic formulation is still the same. It must have SDS, it must have some detergent, so it creates that current and it runs through the system. The rest of the little formulations is besides the point. When you're preparing and loading the samples, you have to make sure of 3 different things in the preparation process to make sure that they resolve the best. One is to make sure, obviously that you have good quality protein extract, but once you have equalized the proteins, right, have equal amount of your total protein in each one of your samples, you want to add SDS to coat the proteins in this negative charge and help to denature them. You want to add beta-mercaptoethanol or DTT, right, which is a reducing agent, and that's going to separate out all those, hydrogen bondings and all those cross links so that the proteins are linearized. And you have to make sure to heat them to help this process out so you can have nice, clean samples. Then once you put them on your gel, they are going to separate from negative to positive end because SDS is going to put the negative charge on all your proteins. And because the smaller proteins will have an easier time going through the pores, the smaller proteins are going to be at the bottom. They'll move faster. And the larger proteins are going to be towards the top. And so at the end of the day, you're going to have decreasing size from top to bottom. So the gel goes from negative to positive. Now in the Western blot, you don't just stop there. You then transfer your proteins from the gel onto a membrane. And that part is called the electroblot or the transfer system. And in the transfer, you can do a tank transfer where you have buffer all over, or you can do a semi-dry transfer like we did in the lab. Either way, the basic principle remains the same. It's still going to go from negative to positive because remember, all the proteins are negatively charged. They were in a buffer running in the system right up here. The buffer was negatively charged, had SDS in it too. The proteins had SDS too. So all the proteins are right now negatively charged. So what we're going to put them on is towards the cathode, the negative side. Then you have your filter paper and then the gel. And after the gel, immediately on top of it will be the membrane that you're transferring your proteins onto. The membranes have charged properties so that it will bind to the proteins and hold on to them when they move through or try to hold on to them. If you can run the electric current long enough, it will go through, but it is going to attach to them. It is going to bind to them to some extent. And then you have the filter paper on the other side and the positive end. So the transfer system, either way, the transfer electroblot also moves from negative to positive. Both the running as well as the transfer runs the same way, negative to positive. So we transfer our system, Now, the membrane itself, there are actually three different membranes that people may use. One of them is nylon, but nylon is actually not appropriate for proteins. Nylon is more appropriate for DNA samples or DNA protein mixture. What we normally use for actual Western blots is nitrocellulose versus PVDF membranes. Um, Nitrocellulose has a lower binding capacity, but and it's a little bit fragile, so you can't keep it for a long time, but it's great for most proteins. It is very well suited for chemiluminescence or a fluorescent signal. It can still be reprobed a few times before it, dies out. And the biggest advantage of nitrocellulose is that when we are done transferring, we can actually look at the quality of transfer by staining it with fast green or ponzo, which stain all your proteins and show you that you transferred good quality protein, that it looked fine, there were no bubbles, and that's especially good for students or early scientists because we make mistakes and we want to make sure that we know if there's a mistake there so we can adjust what we need for it. On the other hand, PVDF can bind a lot more protein. So if you have a protein, your favorite protein is really, really, really low abundance, then that's what you want to use because it's going to bind more. So you are going to have a better chance of seeing it. Same for pore sizes, it's much stronger. So if you have really precious samples and you want to publish with them, and you have perfectly good quality samples and you don't want to lose them and get the most out of them, this is what you would like to use. And what that's going to do is it's going to transfer them and it's going to be very strong and you can just store it in a sealed container for even years, 2-3 years. So if at the end of the second year you're ready to publish and the reviewer asks for additional blot, you don't need to waste time making new samples, which could take months or even years. Instead, you already have them done. You already have the gel ready, not gel, the membrane ready. All you need to do is rerun a Weston with a new antibody and you're ready to go and you can resend the paper. So it saves you a lot of time for those precious, really, complicated samples. So this is something very important for that. We used a nitrocellulose membrane. This is why we must do, especially early scientists as well as students, the transfer check is essential for them. I like to do the transfer check with ponzo stain because it, again, stains better. It has a lower, it can detect even lower concentration proteins, right? It's higher sensitivity than fast green. What are you looking for? This is a good blot. It has proteins at same level of intensity from top to bottom, meaning the transfer was perfectly done from top to bottom all the way. There are no bubbles, so the whole gel and membrane were really nicely in contact with each other. There was no space in there that could leave air bubbles, so the transfer would be incomplete. The markers are very clearly visible. And look, same intensity of protein staining in all samples, meaning the samples themselves were loaded equally. They were all good quality, same quality, and you see nice clean concrete bands. Great blot. Here is another blot, more like what you would see generally. This is very hard to get, and unless you are very experienced. Here is one where most of them are relatively equal, right? You still see some of them are a little bit lower, a couple of them are a little bit higher. This one seems a little bit more high than all the others. So this one will need some playing around to make some conclusions with. More importantly, look, there are a couple of bubbles in one of the lanes. Now, if there are bubbles in one of the lanes, right, in these two, in just this lane, can I use this? So I want to compare these 6 samples, and I want to compare my favorite protein level of expression in these 6 samples. They are probably different, like untreated and low, medium, high, and combination with something else. Who knows? But I want to compare the level of my favorite protein in these 6 samples and make some conclusions out of them. Can I make conclusions about my favorite protein using this blot? What do you guys think? But what if one is untreated? Can you make anything if you don't know the starting level?
Then no.
No. Okay, so let's say, for the sake of argument, I'm just going to annotate a little bit that this one is my 100 kilodalton, right? I can't write apparently. So this is, let me do text actually instead of annotating. So let's say this one is 100, the third band, okay? And the one underneath it is 75 and 50, okay? And the one underneath is 25, okay? So I know they're not quite lined up, but they're kind of sort of lined up. That one was right here. This one is 25. Okay, let's say that my favorite protein was 75 kilodalton, right? So right here, it's gonna fall right in here. Can I use this blot to look for that favorite protein? No. What if it was at 50 though? Can I use it for, or 45 even, let's say 45, so below the 50, right below the 50. Can I? Yes, then I can. So just because there's a bubble doesn't automatically mean I have to rerun the whole thing again, unless my favorite protein falls right where the bubble is. You can still use it for some of the proteins that you might be looking at, right? Similarly, like if I was looking at a protein up here where the red one is, where there is no bubble, I can use it. Or anything underneath, I can use it. But if my favorite proteins fell with the bubbles, then I can't. So that's something to be aware of. Okay. And then over here is something that kind of actually looks like probably they were doing an affinity chromatography type thing, where they have all the samples and then the wash and then your elution fraction, for example. And in this case, they are probably going to probe it now with the antibody against this protein that they were trying to elute off to make sure that this is what they think it is. Okay. Let's see something else. Another thing you can do is looking at the different membranes and deciding which one you should use, right? So we talked about, how PVDF is great for proteins that are kind of low concentration. In addition to that, sometimes, we try out the first time we are running a Western, we try out both membranes and see which one is going to give us a good signal for the antibody we're going to use. So here is an example where the nitrocellulose, you see the signal, but it's kind of faint, right? Hard to see. And when I do the same thing with the PVDF, you see a higher signal. But look here, this is overexposed. This is really hard to make any conclusions with because there is so much signal, right? This protein is really, really easily seen or detected at this using the 0.45 micrometer pore size PVDF. So this will tell me that when I look at this, right, that if I have good quality protein extracts, I can use the 0.2 as well, but then I would need to, again, use the 1 to 10 probably to get better signal. But better to use PVDF with the 0.45 pore size because that gives the best result and shows the most signal. And then I would probably want to use the 1 to 10 diluted sample to get nice clean signal. Here, again, there's another example using 3 different antibodies, right? And I have the samples for PVDF and nitrocellulose. In this case, which one would you use? Would you use PVDF or would you use nitrocellulose? membrane-wise? Which one do you think is going to give you better signal? So for Westons, remember, you don't want merging of the lanes, the signal in adjacent lanes, because it's going to be hard to quantify. And you don't want overexposure either, right? You don't want underexposure where you can't see this the signal, but you definitely don't want overexposure. So looking at these, which one would you choose, nitrocellulose or PVDF? Exactly. In this case, nitrocellulose is going to be a better choice. And would you, in your extraction process, would you use PFA or no PFA? So plus means that they use the PFA and minus means they did not use the PFA. So did they need in the lysis buffer, did they need the PFA or did they not need the PFA to get good signal, to get good extraction? Yes. So for the antibodies they were using for the those proteins did not extract well without PFA. So they want with PFA and they want nitrocellulose. Perfect. The next step, typically after the membrane transfer is to then block. Blocking is extremely important. When we block the membranes, this was the step where we use the blotto. When we block the membranes, what we are doing is we are basically coating any unused site, any site on the membrane that does not have protein bound to it with just some non-specific protein, so you don't get random signal from it. So you don't get this non-specific binding. If you get all these like random bands everywhere, that typically means that you did not block your sample well enough, and now you're getting background noise. So here, again, there are different ways we can block, but in this case, we don't, that's not something we've talked about too much. So don't think about the 2% and 5%, just think about no block and block. So you see the difference between when you did not block versus when you block them appropriately. So if you see a lot of background noise, one of the reasons that can happen is because you did not block, or you didn't block sufficiently at least. Non-fat milk, 5% works perfectly fine for that, by the way, in some of the wash buffer. Okay, next is your choice of primary and secondary antibodies. The primary antibodies is going to target the antigen of interest. So it's going to bind to some place on your protein of interest. If you're just looking for total amount of protein of your favorite protein, right? Total amount of actin, total amount of EGFR, whatever your favorite protein is, then it can target anything, any sequence on that protein. It doesn't matter. The epitope can be along any of that protein system. If you want to look at a specific portion of your protein, let's say you're looking at phosphorylation at serine 32, serine on at amino acid site 32, then you are going to have to use a very specific antibody against that section. So that's the first thing that you want to, first of all, think about, are you looking at a specific section of your protein, or do you just don't care? You just want to know if the protein is there. That's going to dictate which primary antibody you choose. Lots of primary antibodies are present for most of the protein. You have usually different choices. Secondly, you want to think about which animal your extracts are from, or where your extracts are coming from. So if you have mice brain samples, then preferably, you don't always have a choice, but preferably you want to choose an antibody, primary antibody, that was not grown in mice, because that's going to be more likely to give you background from those constant region of your antibody, the IgG section. So those are two things to be aware of. So if I have human samples, I can use mouse antibody raised in mouse. as long as it is targeted to the human protein. If I have mouse protein extract, then I prefer to use rabbit or antibody that is grown in rabbit or sheep or goat or whatever else. Something, a guinea pig, right? Something other than your mouse, because it's going to give better results. If I don't have a choice, then I will still use it, but I will then try to make it very specific to my protein to try to minimize background noise. Now, the secondary antibody is going to be dependent on what primary you're using. If you're using antibody that is raised in mouse, you will use anti-mouse secondary. If you're going to use antibody raised in rabbit, you're going to use anti-rabbit secondary. And it's very important that we are aware of that. I've had times where I'm running like 6 different blots and 2 of them are rabbit and 4 of them are mice, and I've accidentally put them the opposite way, and then I get no signal. Well, I got no signal because I used the wrong secondary, right? It wasn't that it wasn't working, and I would just need to go back and reprobe it, redo the secondary, the correct secondary, and then I will get the signal. Okay. And then the third thing that we choose in antibodies is the type of signal that we're going to detect. The 2 common ones that are used are peroxidase or alkaline phosphatase. Peroxidase or horseradish peroxidase does not usually give a colored product. Their colored product doesn't work well. It's usually a chemiluminescent product. So you have to be in a dark room and do the imaging, or you have to have appropriate imaging technique, right? Imaging detection system to use for it, a gel documentation system to use for it. If you have alkaline phosphatase, you can do it in the chemiluminescent way. You can also use a colored product, create a colored product like we did. Remember, we produced a colored product with it that was visible in that purple band on the blot itself. So we didn't have to do anything else for it. Another thing we can do instead is use a fluorescent tag on the secondary. So you can add like yellow or green or red fluorescent protein on it that just lights up. And then you can have primaries like that too, that already have that tag on it, Alexa Fluor on it, of various wavelengths. And then you can look under fluorescent using a fluorescent detector to look at the signals. For Westons, we typically just do either chemiluminescent or colored product. Very, very rarely do we use a fluorescent system. Now, when you're interpreting a Weston, what are we looking for? We are looking for relative protein expression across the samples present on that one blot. It could be looking at expression levels in different tissue types, could be looking at expression level between untreated and treated or normal and disease, any of those. You can look at protein localization, where the protein is, whether it's in the nucleus or the cell membrane or the outside, or in a condition over time in response to some treatment. You can also look at protein-protein interactions, where we use, for example, affinity protein-like technique called co-immunoprecipitation. We take our protein lysate and we run it through a column with the matrix having our favorite protein on it, and or the antibody against our favorite protein. It's going to capture our favorite protein, isolate it like affinity chromatography, and we will also capture anything that was bound to it. So if our favorite protein is, let's say galectin, and it binds to integrin 1 and it binds to, P53 and something else, it's also going to pull those proteins out. So then when we do the SDS and the beta mercaptoethanol, all of them will separate out. And when they run a gel, we can now find our favorite protein and anything that was bound to it, the other proteins, and we can look for them using specific antibodies against them. Okay, so how are we going to interpret it? Here are some examples. First one is a pretty typical example of something that we would do you have your system, two systems, right? So you have your untreated, non-specific control. And then in this case, I am interested, my favorite protein is called SIRT1. And I'm interested in looking at what happens when I remove it from the system, or at least reduce its expression. So I'm using a silencing RNA, okay? And so you have 3 samples, here it's n equals 3. And I'm looking for different things. There are 4 different blots. The first blot that is important before we do anything else is the loading control. We want to look at, so this is going to use a housekeeping gene like actin or GAPDH or transthyretin, anything that's going to be always present, regardless of what's going on in the cell. And we're going to look at its level of expression and make sure that we loaded approximately equal amount of protein in all our samples that are to be compared. So looking at this loading control, do you think that it is equal loading or not? Yes or no? So we're just looking at the relative level of expression in this actin lane, and these band intensity and size. Are they approximately similar? Yeah, they look pretty equal. So that means it's a good blot. We can compare the different samples and their relative expression. Now we're going to see whether or not our experiment worked. So remember, I had some non-specific silencing RNA, and then I had SIRT1 specific silencing RNA. So on this side, I should just have equal amount, the normal amount of SIRT1. And then on this side, I should have reduced or no expression of SIRT1. Is that the case? Did my experimental system work? Did my siRNA, the silencing RNA, work? Did it inhibit SIRT1 expression? Yes or no? Again, we are looking at the band intensity, right? General band intensity and its size. And so they're all going to have the same width, but they will be less bright, and they might be thinner if there is less of it. So looking at this SIRT1 lane, this particular one, the experimental system check one, is there inhibition of SIRT1 when we do siRNA treatment? Yes or no? Yeah, the bands are lighter, right? And they're thinner. So there is some inhibition. It's not 100%. It's not perfect, right? It's not 100%. It's probably about 50%. It doesn't look more than that. But obviously, when we quantify it compared to this, then we'll be able to see it better. Now, we know that SIRT1 affects the P65 system somehow. So the first thing we are looking at is looking at total amount of P65 protein in all our samples. Does it affect the P65 levels of protein, the overall level of total P65 in the system when we inhibit SIRT1? Yes or no? So now we are looking at this lane that says total P65. So we are looking at P65 levels. Does it affect total levels of P65 when I inhibit SIRT1? They look the same. Exactly. So it did not affect the total amount of P65 present in the cell. So finally, we look at one of those post-translational modifications that may be activating or inhibiting the system. And in this case, it's the acetylation of P65. What happens when we inhibit SIRT1? Does the acetylation of P65 increase or decrease? Yes, so there's more acetylation of P65 when CERT1 is present, is absent, when it is silenced. So that's how you would interpret something like this and expect to see something like this, where I ask you which one is the loading, which lane is the loading control, which one is the experimental check, which system, what is the overall conclusion, right? Does it change the total amount of protein? Does it affect the activation? Something along those lines. Here's another example where when we look at this, the first lane is our untreated control, right? Again, we look at GAP-TH levels to make sure that we have equal loading. It's overall about equal, some up and down, but overall fine. And you have your total amount of protein and you have the phosphorylation levels. Total amount of protein remains the same, no change. Phosphorylated amount goes up against the untreated control. And that's also shown quantitatively over here. And you can see that all of them show about equal activation all along compared to non-treated control. And this was an example with the immunoprecipitation, kind of like what I talked about, affinity chromatography. So we were looking at which proteins are interacting with MRE11. So that's the protein that we are using to purify, right? So we used an antibody against MRE11 to pull down whatever comes with it, okay? And we have a flag tag. So MRE11 was present in all three systems. And then we look at two different proteins, nibrin and XRS2. Looking at this, MRE11 was pulled down by nibrin or XRS2? Which one interacted with MRE11? So actually they use a flag tagged because they probably didn't have antibodies against nibrin or XRS2. If you didn't have antibody against a particular protein, you can tag them with a flag. We call it flag with some random sequence and then use an anti-flag antibody against them. So in this case, they use flag tag to pull these two proteins out that they thought interacted with MRE11. And when they run a Western, which one pulled MRE11? Was it nibrin or XRS2? Only nibrin, exactly. And along with it, it also pulled out RAD50, which was another protein that interacts with MRE11 that was known to interact with MRE11. Here are some troubleshooting things. If you don't have equal amount of proteins, that's a loading control issue. You need to go back and rerun the Western using equal amount of protein in all samples. If you have merged bands and some background coming through, that means you have signal saturation. You need to re-expose the blot at a lower time set, right? So do a lower exposure. If you were doing 1 minute, do a 15-second or a 30-second or 45-second. And if you were doing a 15-second, do a 1 second or a 5-second to reduce the signal so you can actually look at it. If you have bubbles, that's a transfer issue. If you have black or gray background or lots of, that means that you were maybe not wearing gloves or you had dirty reagents that transferred protein on your membrane and now you cannot see it well. If you have non-specific bands, that means you did not block appropriately. And if you have weak or no signal detection, if it's weak signal detection, its antibody concentrations need to be adjusted. But if it's no signal detection, it could be incorrect antibody, primary or secondary. Okay. And stop share. That's that. Was that helpful, guys? Hopefully. So that gives you a little bit of antibody review as well, which I know some of you struggle with. Any other questions?
Um, Just making sure, is the report due tonight?
Yes, it's due tonight. I was gonna change the thing for you to turn in by midnight. If you need extra time, that's fine.
Okay, thank you.
Any other questions? It could, if it is coincide, if those non-specific bands are perfectly aligned with the molecular weight ladder. If it's not, then no, right? So you want to make sure if those aren't really non-specific. It's more that you see that it's the same bands that are aligning with your molecular weight ladder. So you say, oh, shoot, my molecular weight ladder seeped into the other lanes, whether it was because it was broken or whatever, right? If the wells were broken. But otherwise, it is an issue of you're using too much antibody, primary or secondary, or you're using incorrect antibody, or you're not blocking. Good question. Any other question? Okay, then. If there are no more questions, then that's all I've got. And we are gonna then, that's essentially all that we had. Thank you guys for putting up with my crazy semester and my sickness and everything this semester. You guys were great and troopers through it all. So thank you for, adjusting along with me. And I hope to see you on campus and around soon. Yeah, so just email me if you want to meet with me or have any other questions. And we can talk, and just expect to see the final pop up in the next day or two. Hopefully by tomorrow night, otherwise, or not tomorrow night, Thursday, or otherwise by Friday. Okay. Thank you guys. Take care. Bye bye.